So that's the Copan. Copan,
This
I am
is
right?
Copan, yes, yeah.
Okay.
Okay, so what are you doing now? It's
Yeah,
sample plating or
yeah.
AST?
So right now I think it'll do AST, so I just load this sample.
Okay, so this is the this is no
Control
This is
panel.
order
yeah.
to this.
Yeah, so all I have to do is hit play.
Okay.
Everything is barcode driven, so everything's barcode driven. So the sample will get picked up. It scans the barcode. It'll send a query to the LMS, and then the LMS responds with whatever type of sample it is. And based on that, the WASP will know exactly what plates to streak out, what loop to use. So there's a 110, 50 microliter loop. There's a spreader loop for the longitudinal. So you'll see right now it's plating on the 1,000 So it'll go into the sample twice. So it takes a 30 microliter loop. So right now this is the incinerator. So there's an incinerator on board. So it'll sterilize the loop before exactly. Yeah. So once it sterilizes, it'll go into the tube. There's also a camera on board, so it'll take a picture of the loop before to make sure the loop is perfectly aligned, there's no debris. Once it goes inside, it'll take a picture again to make sure there's actually an inoculant in the loop as well. So it takes a quick picture, and then it'll first inoculate the plate. And then you'll see at the other side it's a spreader. So after it goes in the second time, it'll start streaking it for the longer girl.
Okay. Now it is bringing the second sample.
Yeah, so
Okay, yeah.
that way it's continuous and no delays. So the plate will go and get its label taken, and then it will come down the track.
Okay.
So it gets its label on the side, then it gets
Okay,
placed
now the
on
plate's
track.
coming this side.
Yeah. So then, because everything is barcode driven, it gets its barcode scanned, and it'll know, okay, I need to come to the radiant to get a Kirby Bauer disc. So this can hold 50 discs at a time. There's a camera system on board, so it'll check the antibiotic discs and the name to make sure it's placing the right antibiotic disc. In the background, you can program your Kirby Bauer panel, so for your different right, positive and negative organisms, it knows exactly which discs to place. So you can see right now it's plating
What maximum 6 6?
maximum 8 actually. But yeah. But it's best to play 6.
It's plating all of them.
So everything is barcoded and
Yeah, everything's barcode driven, so that way you know exactly where all the samples are. You can always track through our web app system. You can continuously track the samples as well.
Okay, now
So
where
once it's
it
done-
is taking?
Yeah, so once it's done,
All of them, they are coming now here.
yeah, so we don't have the incubator running, but this is the imaging module, and this is a single
Okay.
incubator. So the imaging module holds the camera, so you can program it to take a time zero image, and then you can program it to take whatever other images you want. So if you're doing, say, a 16-hour incubation, it'll know, okay, after 16 hours, let's take it out of the incubator and have its image taken. So that's a single incubator. It holds 795 plates.
Oh.
And then we offer double incubators as well, which will hold double the capacity. Once the image is taken, so you can see that's just kind of an example there on the screen.
Like
so
this,
once the image
okay.
is taken, the user, so the technology is what we call the web app. Then we have something called Matrix. It's our AI module. So it'll actually analyze and interpret some of the culture growth text. That way they don't have to do it and sort through kind of all the different media. So it saves them a lot of time.
Yeah, that's great. Thank you.